that, so far we've got the Th0 cells, we've got Tregs negatively interacting with T helper 2 cells, and those cells, when in excess, produce asthma. So the connections between Th0 and Th2s are both positive. These are not genetic interactions, these are cell interactions. Th0 cells stimulate regulatory and Th2 cells. And the gene that we talked about last time, FOXP3, is important in that production process along that arrow between the Th0 cells and the Tregs. So when you have FOXP3 gene expression, that helps Th0 cells produce more regulatory T cells. So you have high FOXP3, you make lots of T regulatory cells, and those lower the number of Th2 cells, so you get lower symptoms or no symptoms of asthma. And then what did the study we were looking at last time hypothesize? What was their hypothesis about this process related to epigenetics? Okay, so lack of T regulatory cells correlates with asthma, and we saw a figure from this study that showed that in kids from Fresno that had asthma, their Treg cells were not as effective or efficient at doing their job as Treg cells from, say, kids in Palo Alto. We're talking about epigenetics, which means we have to be talking about a gene. There's only one gene up here, which is FOXP3. So what was the hypothesis about epigenetics and FOXP3? So if the histones that contain that gene, somebody look up, where is the FOXP3 gene in the genome? What chromosome? Or everybody look it up. And while we do that, do you still have a question?
Yeah. From the helper cell to the Th2, wouldn't it be suppressed? Like, why would it be activating? If the Th helper activates the Treg and the Treg suppress the Th2, then the Th helper wouldn't suppress Th2?
Well, T helper 2 cells are related to T helper 0 cells.
Yeah.
So this is how it works. Okay. Strange, yes, but this is the situation. FOXP3? Is it on the X chromosome? We've got consensus. How did you do that, by the way? How did you find Google? Okay, are those web pages that are ones we can trust? Gene cards, Wikipedia. Let me see. So, FOXP3. Did you type in anything else or just FOXP3?
Location of FOXP3.
Location. How about human? Make sure that we're trying to find our FOXP3 gene. Okay, so this is, don't tune out. I know this is a little bit distracting, but it's really important, especially because we've got tablets and we've got access to the internet, and we can figure out whether or not we're looking at good genetic data or bad genetic data, that is made up stuff or real data. So which of these hits is the best one to follow? Wikipedia? Okay, so there's a few different things going on here. We've got, this link says ncbi.nlm.nih.gov, National Institutes of Health of the United States, nih.gov. That's a good quality resource to look up genetic information in. If you ever want to know anything about a gene, you can trust NIH sites. What's the bottom one? Looks like a publication in a scientific journal, the European Journal of Immunology. Sounds immunology, Immune cells, we've been talking about these T helper cells, T regulatory cells. That seems like a good place to get information. What about Wikipedia? Why not?
Maybe not. Depending on what the citation is at the end.
Depends on who wrote it. Okay. Where here do we find that information? Oh my goodness. Here we go. Bottom right, location. It says UCSC, University of California, Santa Cruz. Chromosome X. So this I trust. And you don't necessarily know that, but I know that because UCSC has for a long time been a major player in the human genome assembly, human genome sequence. So that's a trustable source. So chromosome X. So the point of all of this is we can make a more specific hypothesis. Sorry, I opened this over that. So what we're suggesting is the X chromosome, essentially, is what we're saying, heavily methylated. What happens to the X chromosome if the chromosome itself gets methylated? What does that epigenetic change do?
Condenses.
Condenses, right? Methylation of the DNA or histones, either, will make the X chromosome this is not X chromosome inactivation, but it will make the X chromosome condense and turn off transcription. What happens if transcription, so that's low transcription of FOXP3. So if we have reduced transcription of FOXP3, what happens to the Treg cells? They're not made. So if you don't have FOXP3, you don't make as many Treg cells. And if you don't have very many regulatory T cells, then what happens? You get lots of Th2 cells because they're not repressed, and therefore you get lots of symptoms of asthma. So that was the hypothesis of this study. That maybe there's something epigenetic control. We breathe in bad air, and somehow that signal, particulate pollution in our lungs, filters down somehow to the immune system and to the methylation state of the FOXP3 gene on our X chromosome.
Current research doesn't know which particular particulate is it.
I don't think anybody knows yet which type of pollution causes this, if at all. So we looked at this last time. This is the data from last time. Regulatory T cell function. The correlation that's positive between regulatory T cell function and whether or not, A, you live in Fresno, which is kind of weird. It seems like if you're in Fresno, if you have asthma or no asthma, your Treg cells are doing worse than if you live in Stanford, Palo Alto, S. But it's always the case that people that don't have asthma have better functioning T cells for where they live. Why might this be, by the way? Does anybody have any hypotheses about why where you live might change how your regulatory T cells function? So geography, so it's a geographical difference. Fresno versus Palo Alto.
Environmental pressures, right?
Well, this says, I'm in Fresno, I don't have asthma. So I'm personally right here. If I move to Stanford, will my regulatory T cells improve? Is that what this says? Not necessarily. So what is the difference then between Fresno population and the Palo Alto population?
What about if the gene expressed is more common here in the valley than it is?
Okay, so if it's genetic, what's the difference between the population in the Central Valley and the population in Palo Alto? Is there a genetic difference generally between the Fresno population and the Palo Alto population? Could be, probably. What would that be? Demographic differences? Is there a racial difference between Palo Alto and the Central Valley? So part of this could be the specific genetics of different human populations. There might be specific genetic differences in Fresno compared to Palo Alto, Stanford, that cause that sort of variation. But even within a population then, this is still showing what we already know, that when you don't have asthma, your Treg cells are pretty good, but people that have asthma have it worse. Lower regulatory T cell function. And that's why I asked you then to make a prediction of what happens to methylation of the FOXP3 gene. So there's one. About a third of you participated, which is better than it could have been, I suppose. There's another. And I got roughly equal numbers of both of these sorts of submissions. What's the difference? Right, so we've got a positive relationship or positive slope, and we've got a negative slope. So FOXP3, we're going to figure out which one it is that's most accurate. So what did we just say a couple slides ago? We were talking through the process of if the gene FOXP3 is turned off by methylation. That's up here at the top of the graphs. Yeah! So if there's high FOXP3 methylation, how should that correlate with asthma? High methylation means what? Yes. More methylation means asthma symptoms. And that's because more methylation means less FOXP3, which means less T regulatory cells, which means less control over those asthma-producing cells. So which of those graphs gives us that relationship? Bottom right, bottom left, or top right? Bottom left. Where we've got high levels of methylation would correlate in individuals in Fresno that have asthma. And a number of you did submit graphs that looked something like that. So within asthma, or within Fresno, if you have asthma, you might be expected to have the highest levels of methylation of that gene, and then decreasing there to Fresno with no asthma, Stanford individuals that have asthma, and Stanford that don't have asthma. And that's exactly what they saw. So these scientists looked specifically at the methylation state of the FOXP3 gene. And this specifically is methylation, as we've discussed before, of those CpG motifs, that's C's and G's next to each other with the phosphate backbone in between. Those are the ones where the cytosine gets methylated. So that's what they were looking for. They were looking for C's and G's next to each other on the DNA sequence near the FOXP3 gene. They find that in Fresno, if you've got asthma, your gene is more heavily methylated than somebody who's living next to you, or a sister, or your parent, or whatever, that doesn't have asthma. Chemical differences, chemical modifications to your DNA. And a pretty sizable difference between people that have asthma and people that don't in the same geographical location. So in conclusion, somehow the environment impacts gene expression. Asthma, methylation, low FOXP3 production, asthma. The good news is epigenetic marks can be changed. It's not like mutations where you make one change to a nucleotide and that's it. Methylation marks can be removed. And acetylation can be removed. So histone modifications, bless you. Presumably, taking yourself out of the environment could change things. So if you're in Fresno and you move to Palo Alto, that doesn't mean that you're always going to have asthma. It means that maybe if you chat 5 years from now, your methylation status of your FOXP3 gene will change. Oh, okay. Yeah, that's the beautiful thing about epigenetics is that they are sometimes heritable, but they're changeable within an individual's lifespan. Whereas we can't do anything about our DNA sequences. Yeah.
They suspect that there's correlation between that. If you are removed from the environment, how about case studies where they stay in the same environment, but they no longer have the symptom or the asthma as an adult that they had as a child? So I know there's a few case studies of where that is.
So what's going on when you have a single individual that doesn't change environments, but their phenotype still changes? A whole laundry list of things that could be happening there. Unfortunately, it's hard to pin down from a basic story like that, I suppose, what might be happening. You could be having a different environment, you don't move physically, geographically locations, but there could be other lifestyle changes, for example, that would cause gene expression changes, epigenetic change. So what they don't know yet, and I know we're a couple minutes from done, which is good because I'm a couple minutes from done. What we don't know yet is how actually pollutants in the air getting into our lungs goes through several steps probably to get to changing the methylation state of the X chromosome. So what I want you to do, a few things. There's another survey that I've added today. This one's only got a couple of questions, I think, if I remember correctly. So as it says here, it's the URL's on Blackboard now. Please complete it by the last day of instruction. This is a fantastic cartoon. I love this cartoon. You want a back scratcher? You do this whole thing, right? Easy way to make a simple device. What are these called? See, this general idea of making a really complicated thing to do something really simple. They're Rube Goldberg machines. So what I'd like you to do, so these are a few things for next class. I'm going to open up a new Google Classroom assignment. What I'd like you to do is invent a gene expression Rube Goldberg machine that somehow takes pollutants floating around in the air, getting into your lungs, and then resulting ultimately in methylation of the FOXP3 gene, giving you asthma. So we've talked about some specific steps already. I want you to fill in the blanks. So here on the bottom, you live in Fresno, pollutants land in your lung cells. Fill in something here, draw it out like a Rube Goldberg machine cartoon that leads in the methylation of FOXP3. Lastly, one more movie for next class, Sex Determination, and then you can read those bottom points yourself. Okay. If you've got any questions, come on up. I've got a couple of minutes to talk, and have fantastic weekends.